Hello, and welcome to this Pharmaron podcast, part of our DMPK Insights podcast series. My name is Scott Summerfield, and I lead the Metabolism group in our UK-based integrated DMPK discovery and development platform. Today, I'm delighted to be joined by Professor Elisabeth de Lange to discuss the evolution of CNS drug disposition models, taking a brief tour of the past, current state, and what the future might hold. Welcome, Elisabeth.
Yeah, thank you so much. I'm very happy to have this podcast with you. That is very nice.
By way of introduction, Elisabeth is a professor in predictive pharmacology and principal investigator at the Research Division of Systems Pharmacology and Pharmacy at Leiden Academic Centre for Drug Research, abbreviated to LACDR for the rest of the podcast. With her team, she is helping to unravel the rate and extent of mechanisms that govern central nervous system, target site pharmacokinetics, and the related pharmacodynamics, with special emphasis on physiologically based PB translation between species and conditions. Ultimately, this is to have a mathematical model that supports CNS drug development, and that includes reduction and replacement of non-clinical studies, and can predict the best possible treatment for CNS conditions in individual patients. Elisabeth has contributed more than 160 publications to the peer-reviewed literature, over 170 invited lectures, and organised in excess of 100 conferences, symposia, workshops, and courses. She has multiple leadership positions in scientific projects, also at the LACDR, nationally with the MVF, in large international organisations such as APS, and is currently coordinating the EU consortium QS Pain Relief. She has a number of advisory roles, both advice and consultancy, and alongside all of this, Elisabeth contributes to education in the biopharmaceutical sciences, bachelor and master's courses. Among other honours, Elisabeth received the AAPS Fellow Award in 2013, an honorary doctorate in pharmacy from Uppsala University in 2020, and the prestigious Shiner Lecture Lifetime Achievement Award from ISOP in 2020. Also of note here for this podcast is physiologically based models such as CNS PK 3.0 that predicts pharmacokinetics in multiple locations within the CNS of mice, rats, and humans, and is an important example of the topic that we're going to cover today. So again, Elisabeth, thank you so much for the chance to talk with you about CNS, DMPK, and PD. It's fascinating because of the unique properties of the blood-brain barrier and the highly privileged position the brain holds in health and disease. So recently I was reminded of an article published in 1997, co-authored by yourself, Margareta, and Lennart Pausau, where there was a simple two-compartment model for the passage of unbound drug across the blood-brain barrier from blood to brain. And actually probably the first mention of what would become known as Kpuu, the unbound concentration of drug. Fast forward to today, and the modelling efforts are far more sophisticated. And again, a great example being Lange CNS PK3, which describes multiple compartments and fluid flows during the passage of drug along its journey from the peripheral circulation across the blood-brain barrier and through the brain. So over that period of about 30 years, multiple scientific advancements have collaborated, leading to the emergence of PBPK, PKPD tools, and hence the topic for this podcast. But first off, can you describe what kindled your interest in modelling and helping to answer key questions around the drug disposition into the brain?
Yeah, sure, I can. So, of course, there is a driving interest in trying to understand everything that governs blood-brain barrier transport, and also what goes beyond in terms of pharmacokinetics in the CNS itself, in the central nervous system itself. And looking at that, we know that it is rather complex, and there are many processes ongoing that are kind of interrelated. So that means that one location PK may differ from the other ones because local pharmacokinetic processes might differ as well. And we just cannot put that in a graph in a 2D way or in a 3D way because it's actually multidimensional. And to that end, you kind of need advanced models, mathematical models, to put these all together. And I started with microdialysis as a tool to investigate blood-brain barrier transport and beyond. And I was in a group where there was a lot of modeling. So I also kind of learned aside from the modeling. And with that, I kind of integrated it into the microdialysis work. And ultimately, with the modeling, I visualized a CNS pharmacokinetic model that would be like a TomTom navigator. Because so this navigator, you put in, what kind of traveler do you have? Are you a bike? Are you a car? Are you a boat or a train? And where do you want to go? And what will be there on your route? What traffic will you go? So what you need is an infrastructure. You need to know what is the traveler, and you need, and the outcome is the resulting traffic. So that kind of relates to the CNS infrastructure, the drug that is traveling, and the traffic is actually the pharmacokinetic. So I thought, okay, so how did they work? So of course they needed a lot of input on their development of that model, and they needed time and location specific traffic. And with that kind of the prediction came. So that was kind of how things got sparked.
Well, it's a nice analogy, actually, the traffic one. I've not heard that before, but it's useful to kind of contextualize. Thank you.
So but everyone can easily understand because they use it, right?
Well, yeah, and absolutely. And the thing is, it strikes a point about, mathematical models in a sense are quite abstract,
Yeah.
So being able to contextualize that for, as an introduction is obviously really valuable. And that's great. Thank you.
Yeah.
So in terms of understanding the relationship between unbound concentration and effect, brain Kpuu is now a well-established parameter, and like I said, that 1997 paper was one of the first kind of mentions of unbound concentration gradient across the blood-brain barrier. But there's another important part of the story which I took from your bio sketch, and namely that's contextualizing both rate and extent of brain passage and transport. And I know, having been a strong advocate for considering both rate extent by yourself, I wondered if you could share your thoughts on this and the importance of modeling the relationship between concentration and effect.
Right. So indeed, so I think also with Moderator, we teamed up so nicely in so many ways. And also that paper was a kind of very good start of our good collaboration and insights that we on the way gathered together, this piece by her, that piece by me, and we kind of used it over, a crossover way. But indeed, so the Kpuu brain, or maybe mechanistically we should call it Kpuu blood-brain barrier, because it focuses more really on the blood-brain barrier itself. It is actually the ratio of unbound drug concentrations on both sides of the blood-brain barrier, but at steady state. And actually, it is not the ratio at any point in time. So it means that the pharmacokinetic profile of the brain extracellular fluid depends on both, and that is what the receptor sees. So if we have a very simple case of a compound that just passively distributes into the brain, and it has a Kpuu blood-brain barrier of 1. So this is also in the 1997 paper. It could be a compound that is slowly crossing the blood-brain barrier or fast, or everything in between. And it really makes a difference in how the relationship is between the unbound concentrations in the brain extracellular fluid relatively to the unbound concentrations in plasma. So it means that so the receptor or the target does not see a Kpuu, it sees the concentration as a function of time. So that is what we need to take into account. And that is why I believe that also we have to address rate as a very important one. So the 1997 paper was actually the thinking beyond. So it is both rate and constant.
Yeah, thank you, Elizabeth. And I know there's papers such as the Mastermind one that you did subsequently, which elaborate a lot of this, and they're a fantastic read for those listeners out there.
Yeah.
So one approach that I've seen to describe how a model breaks down into 4 categories, and it's kind of a loose thing, and maybe we can just talk around it today, is firstly whether a model is descriptive, so trying to understand what's already happened, diagnostic, trying to explain why we've seen it, predictive, what could happen, and then prescriptive, what should happen. And just going back to that 1997 article, and I'll mention the name here so people can go look at it. So it's drug equilibration across the blood-brain barrier, pharmacokinetic considerations based on the microdialysis method, and that was published in the Pharmaceutical Research Journal. So at the time, that was arguably a descriptive model focusing essentially on assessing brain pharmacokinetics at the level of the BBB. So where do you feel that we are now in terms of modern models like CNS PK3, I guess with respect to being descriptive, diagnostic, predictive, or prescriptive? And then before that, the kind of key developments that took us from those very simple models to where you think we are now.
Right. So I like very much your kind of categories of what a model could be, or how to describe a model. Actually, for the 1997 paper, it was not actually descriptive because it did not fit data. It was actually more a theoretical approach to understand how rate and extent of blood-brain barrier transport would influence the relationship between brain extracellular fluid pharmacokinetics and plasma pharmacokinetics. Of course, the unbound should always be the unbound. And it also included the so-called what-if scenarios. So if a drug is passive with regard to blood-brain barrier transport, if it's passive only, active in flux transport, active efflux transport, or combinations of that, what could we see? And actually with that, it gave kind of insights if what could happen. So also, and of course, the model was elicited by a discussion between Moderator and myself on the microdialysis data that I produced, because that was new, because CSF was the side of sampling before. And so it provided also new information on how things could work. And with the theoretical approach, actually, it could also kind of explain what we have seen with microdialysis, and also a little bit what could happen, and but not yet what should happen, because it was too simple at the time. So it was really revolutionary at the time, of course, but then we got more insights, etc. And with the Lange CNS model, we actually also included not only what happens at the level of the blood-brain barrier, but also beyond, in terms of what is the bulk flow of the extracellular fluid, what is the extracellular distribution, what is the CSF flow, and all these kind of things, and also in terms of the physiology as it is. And with that, we could actually have information on pharmacokinetics that is indeed CNS location dependent on the relative contributions of the pharmacokinetic processes around that location. It can clearly show drug dependency, and also a drug property can be put in to see what drug property would lead to what kind of pharmacokinetic profiles. And also it clearly shows the species dependency, because it's ultimately based on physiology that you can scale. You can scale the physiology, and the drug remains the drug, whatever subject you give it to, or whatever situation you give it to, but the physiology, so the context, is the different one. And what was very important to that end is in developing is also that you have data that shows the interaction of the processes within the same single subject. So now I touch a little bit upon the mastermind approach published in Fluids and Barriers of CNS in 2000, what was it, 10 or so? But that we had so-called smart data, measuring with microdialysis not only in one spot in the brain extracellular fluid, but also in different locations in the CSF, and kind of combine all that information to tease out with mathematical modeling what should be the interrelationships, and then do that for multiple drugs, and then in a very structured way have kind of the general principles of drug distribution across the blood-brain barrier and into and in the CNS itself.
Thank you. Obviously, there are two, in any model, you've kind of got drug-specific parameters, and then you've kind of got system-specific parameters, and you kind of mentioned all the flows and things, and they're
Yeah.
Obviously really important in those differential equations that are really describing what happens. I guess there's quite a lot of fundamental research besides the computing capability to kind of build that system specific data set up across those three species. And I guess did you draw on literature and maybe experiments as well to build that information up?
Yeah. So the key technique was actually microdialysis, and we did it in rats. So that was the species that we built up all the information. We also did microdialysis experiments in mice, in earlier stage and also in a little later stage. And of course, in human, we do not have, or only scarcely have, microdialysis data. So that was also the Margareta's group that actually had the results of morphine, for instance, in TBI, traumatic brain injury patients. But so microdialysis was key, but also for all the physiological information, you need to have a picture of what the brain is. So the brain or the CNS infrastructure. And I made a kind of a drawing of that, and a very kind of a block here for a cell and a blood-brain barrier here, blood-CSF barrier there, and then flows, etc. Just to visualize the map of the CNS, and then collecting indeed information on surfaces, on flow, and then in different species. So now we have mice, human, and rat. We're working on the pig as well at this moment in time. So trying to also have this bridging possibility, because if you know the physiology of a certain species, you can bridge it to the physiology of another species, and then with that bridge the pharmacokinetics, and that makes it ultimately a very strong way to be used also across species, across drugs, across conditions, etc.
I guess back in 1997, I can't even remember what computing was like back then.
No, it was rather simple, actually.
Yeah.
And if you also see the differential equation, it was advanced at that time. And it was really, oh, wow. So that type of feeling, okay, we can kind of start to understand how things might work. And then was so interesting. And that was with Berkeley Madonna. That was kind of a software where we did these things in and also explored many other things. It was, much simpler at that time. And now, of course, it is a circus of differential equations and all kinds of everything, but it's a step by step with that. And also we are kind of very deep into all aspects of the CNS as far as we can get.
Just reflect on a bit of the stuff that we've just covered and thinking on modern CNS models and those advancements. What do you see as good examples of major achievements of what's now possible in models like CNS PK 3.0 and others that are trying to do the same thing in terms of CNS PK and PK/PD?
Yeah, so it is actually, of course, it is the scientific community with CNS PBPK modeling and PD modeling that all together went to the next stage in the last years. And well, there is so much more possible. So if I look at the Lange CNS model, because I can talk on that easily, of course, we can kind of see, or we were able to adequately predict the very scarce data from human. That is also the reason, because we cannot just sample it. We can, and we can predict, and that makes it much stronger. So if that is adequately done, even if it is CSF, that is not the target site. So we can see, okay, if CSF is okay, we gain trust in also the predictions of brain extracellular fluid. And with that, we come to a next step. And of course, it is the concentration at the target site that drives the pharmacokinetic. Yeah, so I think with now, so what we did, for instance, was see, okay, what would change for small molecules in terms of drug distribution, for instance, for Alzheimer's patients. And the interesting thing of that was that actually we predicted for the 3 drugs that we investigated that we did not predict changes so much. And that is also an interesting thing. And then there were also experimental data in mice, in Alzheimer mice that showed the same thing. So there was, for small molecules, the ones that have been investigated, there was no change. So with that, for instance, we learn, okay, what has been published about all kind of changes in Alzheimer's disease at the blood-brain barrier should merely reside on other mechanisms, not not governing the small molecules. So not so much the efflux and the influx and the passive things, but merely the vesicle-based transport modes. Yeah? And if you look back, for instance, horseradish peroxidase, for instance, is also not just blood-brain barrier, as we call it all blood-brain barrier permeability, but of course we have different aspects of that. And horseradish peroxidase is also actually vesicle-based transported. So that is interesting. So with that, you can also kind of tease out what is going on. So let's see what was your question remaining. So what is now possible? Yes, what is possible? So we can, in a way, predict for any drug molecule where we do have, of course, the pharmacokinetic input. So pharmacokinetic in terms of plasma. So what kind of profile would we expect there? And that is also where we can use other PBPK models to see, okay, if you have this dose regimen, what would be the resulting plasma pharmacokinetics? And that is used as an input to expose the CNS to, because we have a CNS PBPK model. And then we could see, okay, what would we expect for a certain drug molecule how would we expect its pharmacokinetic behavior? And that could be related to the need for what concentrations, or the information for what concentrations would be needed to stimulate a receptor or to have a proper interaction with the target, etc. So that could still be done in vitro, but also in vivo, we can do a lot of things. So, well, you mentioned in the introduction, indeed, I'm chairing the European Horizon 2020 QS Pain Relief Consortium. And in that, we really try to integrate different models in sequence. So we have the CNS PBPK model. Then we have a CNS binding kinetic model. And then we have neural circuit models. So it's, of course, with the consortium partners. And we have pharmacodynamic data at the level of in vitro, so cellular aspects. We have in vivo animal work. We have human participants, the healthy ones, and the ones for QS pain relief, of course, the pain patients. And we have the pharmacodynamic outcomes. So we try to, with actual data as well as model approach, we try to integrate, and with that have a picture on what is the pharmacokinetic that drives ultimately the pharmacodynamics. For instance, at the level of analgesia, but also on cognitive impairment, on abuse liability, on all these aspects that come, for instance, with the use of opioids that are kind of central in this study. And with that, we play a kind of Sudoku game. And so I mentioned Mastermind before, but now it is much more complex. Yeah. So now we're playing the Sudoku game. And we try really to tease out what. And so for each part, we try to peel off from dosing to the ultimate effect, all the mechanisms in between. And we have come to the receptor occupancy. And we want to get beyond that, of course, to really see what is between receptor occupancy and the ultimate effect. And so, for instance, cyclic AMP changes and all these kind of things that can be derived from in vitro. So with that kind of, constantly progressing in our understanding and in our predictive power of ultimately, and that is my ultimate aim, to be able to predict the pharmacodynamics in human ultimately.
I mean, that's really nice applied research, really, because obviously, Paul Morgan and colleagues at Pfizer have put forward, the 3 pillars of drug survival, which, some people now refer to kind of as the 4 pillars, because you've got, the downstream stuff.
And actually, the more you know, the better you can predict, right?
Yeah, of course. And, the attrition in the CNS drug development is really high. It's very hard, and many circumstances to kind of derive an unbound concentration in a human brain without, resorting to, PET gives you total, CSF, there's the lag and the meaning of, transporters flow and everything. So, and then the downstream pharmacology as well that you mentioned. So back in the day when just before 1997, I studied biological mass spectrometry, which now is kind of referred to as proteomics. And part of one of the things back in the day, I would never have imagined was this sort of pharmacoproteomics, where you could look at the levels of transporters in the blood-brain barrier and other regions, organelles, and and kind of begin to use that. Is that something that's how's that integrated into kind of the modeling in the CNS now?
Yeah, we definitely do that. And indeed, thanks to the progress in this mass spectrometry field, so it's really intriguing. We use expression ratios between species for a certain transporter when it is known as one of the translators as well. Yeah? And for instance, if we have a Kpuu in the rat and we don't have it in human, okay, how can we make a translation on that? But also we dive deeper, so we do multiple new things. So there are things that have not been addressed explicitly so far. So that is metabolism for drug metabolizing enzymes in the brain. So that is what we currently work on, and we have data that we now can really capture. So that is nice. Also CSF flow. What would happen if you drug a dose intravenously, but also intracerebroventricularly? Because then it should be the same flow, right? If the drug doesn't change the physiology, that is also what we have to take into account, of course, but let's assume it does not. And now we have, for the red, we also have adapted CSF flows according to what would happen after intracerebroventricular administration. And it should, of course, also hold for the intravenously administered drugs, and it does. So that is also an improvement. Then also brain tumor, but coming back a little bit more on your starting in the question with regard to spectrometry or mass spectrometry, we are also diving into, or we have already made steps with that, with if we have blood-brain barrier and we want to know also on that level and also, of course, for the rest of the brain, but what is there as a transporter? And what is the expression of the transporter, the protein expression, but also what is the relationship between protein expression and functionality for a certain drug? And now we have kind of a sneak preview. We have work that will be published in the coming time, where we found that there is a drug-dependent relationship between the change in expression of a certain P-glycoprotein, in this case, that we took, and the resulting change in activity for that drug. And because we had, at one point in time, studies that were done in about a little bit more than 10 years ago in epileptic animals, where we found in the same animals, and always a very important emphasis, because then the data are connected, we found after induction of a seizure, we did experiments with animals with kinidin, P-glycoprotein substrate, and so we had the profiles with microdialysis, and then we added tarikidar to block PGP, and then we found, and we just at different times after the seizure, so different animals were kind of used for 1 day, for 2 days, 3 days, 4 days, up till 30 days. We also measured in histology PGP expression, blood-brain barrier expression of PGP, and related that to the activity of PGP, and there was no relationship. So there was a huge change in time in the expression, so it went up and it went down, but it was not related to a change in the pharmacokinetics of kinidin being a PGP substrate. So that is also what kind of spark, okay, what is going on? And we now have a handle on that. So that is what the future will bring. And with that also, if we know expression of transporters and we have a little bit more information on, what drug and what is that relationship, we can make next steps. What would we expect in that case? For instance, in diseases, like Alzheimer's disease, P-glycoprotein changes, does it make a difference for a certain drug if we talk about small molecules? Large molecules is a different part, of course. But
Elizabeth, thank you for sharing that. I will wait with bated breath. It's an area, and the CNS is very dear to me from my time in pharma. So that sounds fantastic. So we've talked a bit about sort of that as like an emerging opportunity as well for development in CNS PK and PD modeling. What do you think would be valuable next from a research perspective? And obviously, you've got sort of the very heavy science that happens in academia and also in industry. What do you think are the kind of important things that will really help the next generation of modeling?
Yeah, so what I think, if it comes to the role of pharmaceutical industry, I think there should be a forth and back because pharmaceutical industries have lots of data that could be shared in a way. And ultimately up till, so what has been done in vitro setting, in preclinical animals, what were the first in human studies, to really see everything. And also with what successes, that is what we know of. And then there's a little bit published about things, but not all the data are there. It is just kind of what we know, but also why things did not work. Right? Because we can learn so much of all the things that did not work. And there's so much of data. And I think we can team up. So academia can team up with pharmaceutical industries just to get a handle on why is something successful and why is something else not. And that is also the big Sudoku game again. If you have the people that like doing games like that, to defragment information, to really order it and to put it into a mathematical model that really can explain everything, then I think we are really in the next league.
Yeah, Elizabeth, thank you so much for sharing your thoughts on the current state of the art with models such as Lange, CNS PK3 and others, and what the future might hold. Thank you very much.
Yeah, thank you too. It was really nice to have this conversation with you.
And also for our listeners, thank you for joining us for today's episode of Pharmaron's DMPK Insights Series. We would like to remind you that our DMPK webinar series is also available on demand, covering a variety of key questions related to DMPK science and drug discovery and development. Stay tuned for more podcasts in our Pharmaron DMPK Insights Series. Thank you very much, and bye for now.